And hi everyone, my name is Khairunisa and my matrix number is 99394. Today I'm going to present the data and the conclusion.
Hello and hi, my name is Martuto Hengki Jamiling and my matrix number is 99626. So in this video, I'm going to present about the procedure or method that we do during this experiment.
Hi, my name is Nuratika Mutiaziz, matrix number 100538. In this video, I will introduce to you the materials and the apparatus used in this practical.
My name is Muhammad Haikal bin Muhammad Hanif. My matrix number is 99729. In this video, my parts are objectives and group.
In this experiment, there are several objectives. First is to understand the basic principles of spectrophotometry and how it is used to measure the concentration of proteins in a solution. Second, to learn how to properly operate UV spectrophotometer and handle cuvettes without contaminating samples. Third, to practice preparing a series of protein standards and measuring their absorbance F595 nanometer using the Bradford assay. Fourth, to be able to construct a standard calibration curve and use it to determine the protein concentration of unknown samples. And lastly, to improve lab skills such as pipetting accurately, handling reagents safely, and recording results clearly and systematically.
For this practical, the material you will need are distilled water, 70% of ethanol solution, BSA protein, Bradford reagent, and standard unknown protein solution labeled as A, B, and C. While for the apparatus, you will need test tubes, test tube rack, cuvette, micropipettes, and spectrometer.
This is the test setup for this experiment. Now, let's go to the first part, which is the preparations of the standard curve. First, 6 test tubes were labeled and 4 ml of distilled water was added to each tube using a micropipette. Next, 1 ml of Bradford reagent was added to each tube using a micropipette. A duplicate sample of 50 microliter, 100 microliter, and 200 microliter of the 1 mg per milliliter BSA protein standard were pipetted into the tubes to create a concentration series. After that, the solutions were mixed gently. Now, we go to the cuvette handling. Before transferring the solutions, the cuvettes were checked to ensure they were clean and free of scratches or fingerprints. The mixed solution were carefully transferred into the cuvettes, filling them to about 3/4 of their capacity. The spectrophotometer was zeroed using the blank. The absorbance of each sample was measured starting from the least concentrated and the value were recorded. Now we go to the part 2. In part 2, the same procedure from part 1 was followed. But instead of using the BSA protein standard, unknown protein sample A, B, and C were tested.
For the data, based on the graph that we got, as we increase the protein volume from 50 to 200 microliters, we observe a steady rise in absorbance values, which is from 0.080 up to 0.116. This confirms that the assay is sensitive to increasing protein levels. However, the smaller increase between 100 and 200 microliters suggests the beginning of saturation, a point where the dye cannot bind to any more protein, which is typical in colorimetric. Standard curve shows a strong linear relationship between protein concentration and absorbance at 595 nanometer, following the equation y equal to 0.02x. This confirms the Beer-Lambert law, where absorbance increase proportionally with concentration. Every 10 microliter per milliliter increase in protein led to a consistent 0.2 unit rise in absorbance, making the curve a trustworthy reference for calculating unknown protein concentration. To conclude everything, our spectrophotometry experiment showed a strong and consistent relationship between protein concentration and absorbance at 595 nanometer. As protein volume increased, so did the absorbance, confirming the sensitivity of the assay. We noticed a slight plateau between 100 and 200 microliter, which may be due to a saturation, a common effect in colorimetry assay. Using the standard curve we generate, with the equation y equal to 0.02x.
In the last practical session, our group have obtained several reflections. First, we collaborated to learn how to operate a spectrophotometer for measuring the absorbance of various solutions. Our objective was to construct a calibration curve and apply it to determine the concentration of an unknown sample. Second, working as a team allowed us to stay organized and complete the task efficiently. We split the responsibilities. Some prepared the solutions, others prepared the spectrophotometer, and the remaining members collected data and created the graph. Third, we improved our technique and achieved more accurate results through group discussions. For example, we resolved the issue of bubbles in the cubes, which helped us obtain more reliable readings. And lastly, these experiments deepened our understanding of how spectrophotometry functions and highlighted the importance of precision. We also realized that clear communication and effective teamwork greatly enhance lab efficiency. In future, we aim to improve our time management and pay closer attention to minor details.